Welcome back to Chapter 8, Nucleotides and Nucleic Acids. In this lecture, we will learn about nucleic acid structure and chemistry. Shown on this slide is the original model proposed by scientists Watson and Crick. They proposed that the DNA structure is a double helix, and this specific form of DNA is called as bDNA. Two strands, this strand that is shown here and the opposite strand, run anti-parallel to each other. The interface of the strands, as you can see, is held together by complementary base interactions. For example, adenine pairs with thymine and guanine pairs with cytosine, and they pair by hydrogen bonding interactions. The double helix forms a major groove, as shown here, that is a major groove, and a minor groove in this specific double-stranded structure. Now, to make this more simpler, another stick representation of DNA is shown here. And as you can see, the major groove is here and the minor groove is here. And if you look at the surface representation of the space filling model, it's even more clear. The major groove is larger and the minor groove is smaller. Shown here is another representation of a double-stranded DNA. And here it's zoomed in to view the molecular interactions between two bases. In this case, adenine and thymine, and guanine and cytosine. As I've said before, adenine forms 2 hydrogen bonds with thymine, whereas guanine forms 3 hydrogen bonds with cytosine. Now, the major groove side of the B form of a DNA is on the side that is above both the bases. Similarly, for guanine and cytosine, the major groove is on top of them, whereas the minor groove is on the bottom side, and that is closer to the glycosidic bond or the ribose ring. That is same for guanine and cytosine. DNA strands are complementary to each other. As seen in this image here, the two chains, the one on the left that goes from 5' to 3', or one on the right that goes from 3' to 5', differ in sequence. But they are complementary to each other, that is based on hydrogen bonding interactions, which means A pairs with T and G pairs with C. And most importantly, they run anti-parallel, and hence they are called anti-parallel strands. Now, if you look at the sequence as shown here, from 5' to 3' for the left strand and 3' to 5' for the right strand, they're complementary. CG, AT, AT, TA, CG, GC, TA, CG, AT. So, anywhere you see a double-stranded DNA, both the strands will be complementary to each other. DNA can adopt 3 regular superstructures. As shown in this figure, there are 3 different forms of DNA: the A form, B form, and a Z form. Each structure shown here has 36 base pairs. And in here, the blue color is the phosphate backbone, and the yellow colored molecules that are shown here are ribose and bases. The most commonly found form is the B form, and it is found in cells. A form, which is shown here, is found in nonpolar environments and is more compact as compared to B form and Z form. Z form, however, is more extended, as can be seen here, and can be found very infrequently in cells. This table summarizes some properties of these three forms of DNA, and it is good to remember some of these properties. For example, the helical ends for both A and B form are right-handed, while for Z form is left-handed. The helical diameter is the highest for A form with 26 angstrom, while B form is 20 and Z form is 18 angstroms respectively. Base pairs per helical turn is 11 for A form, whereas 10.5 for B form and 12 for Z form. Another important point is the glycosyl bond conformation, which is the same for all three forms for pyrimidines, anti, and anti for pyrimidines, whereas it's syn for purines in Z form. And that also contributes for it being a left-handed double helix. So why is the double helix a big deal? Two reasons. The first one is that the double helical DNA allows for a high fidelity replication of genetic information. Secondly, each strand can serve as a template for new complementary strand formation. Watson and Crick, in their original Nature paper, wrote this: "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." So they had originally postulated one of the applications of this double strand in a DNA. During DNA replication, two parent strands become part of two different daughter strands. So how does DNA replication happen? First, strand separation takes place. Next, each strand serves as a template for the synthesis of a new strand. So for example, this strand that comes along for the first daughter strand on the left, and the complementary strand comes along as a template for forming the daughter strand on the right. Synthesis of the new strand, as shown here in red color, is catalyzed by enzymes known as DNA polymerases. Newly made DNA molecule has, as you can see, one daughter strand and one parent strand. And this is precisely how DNA replication happens. And this can, in fact, store genetic code. Not all DNA is found in simple double helices. There are certain sequences that have highly stable secondary structural features, such as palindromes and mirror repeats, as you can see here. Palindromes are sequences of double-stranded nucleic acids with twofold symmetry. To superimpose one repeat on the other, it must be rotated 180 degrees about the horizontal axis and 180 degree above the vertical axis. So TTAGCAC, if you rotate it about the horizontal axis and you rotate it about a vertical axis. So horizontal axis is rotating it like this and vertical axis is like this, and then you will get this shaded portion. So here, as you can see, TTAGCAC is palindromic to this sequence, which is CACGATT. Now, every time you have a palindrome, you have a reverse complement that follows that specific sequence, which means that C is complement to G, A is complement to T, C is again complement to G, C is complement to G, A is complement to T, and both A's are complement to T. So it's literally a reverse complement of the sequence that one writes. A mirror repeat, however, is straightforward in the sense that the sequence that is written here has a symmetric sequence with each strand. Superimposing one repeat on the other requires only a single 180 degree about the vertical axis. So the vertical axis is this, and you rotate it about 180 degrees, this specific shaded sequence, and you will end up with this sequence. Palindromic DNA is commonly found and can be on the same strand or on different strands. When a palindrome is found on the same strand, as can be seen here, they usually form hairpins because a palindromic sequence can hydrogen bond with another palindromic sequence. For example, TGCGAT can hydrogen bond with ATCGCA, and hence this strand becomes a hairpin. When on different strands, palindromic DNA is often an inverted repeat, as shown here. Here there are two strands, and each strand has palindromic sequences. And in this case, this forms what is called as a cruciform structure. Each strand, the palindromic sequences in each strand hydrogen bonds to themselves as compared to hydrogen bonding on opposite strands. And this happens to be a thermodynamically stable structure, and it allows pairing within the same strand in an alternative structure like this. Nucleotides participating in a Watson-Crick base pair can form additional hydrogen bonds with the third strand, as shown here. This is the usual Watson-Crick hydrogen bonding between adenine and thymine, as shown here. The adenine can also form additional hydrogen bonds with another thymine molecule. And in this case, the N7 of adenine and the nitrogen at C6 position can donate hydrogen bonds and accept hydrogen bonds, resulting in what is called as a Hookstein pairing or a Hookstein hydrogen bond. Similarly, GC structures can also form hydrogen bonding. In this case, guanine as hydrogen bonded with cytosine. In Watson-Crick hydrogen bond, there are 3 hydrogen bonds as seen here, and it can also form Hookstein hydrogen bonding with a cytosine. The 2 hydrogen bonds, one is accepted, 2 of these hydrogen bonds are accepted by cytosine from this protonated cytosine. As you can see here, this NH, this nitrogen, is protonated in this case. And one of the very reason why this nitrogen is protonated is because the pKa of this nitrogen is usually increased, and at physiological pH, this cytosine that is involved in the third hydrogen bond is protonated and hence can have a hydrogen on it to donate it to a guanine. Hookstein pairing, as shown here, can allow for the formation of a triple helix, as shown here. The triple helical DNA containing two pyrimidine strands are shown in red and white. In this case, the blue colored strand is the purine strand. This forms a Watson-Crick base pair with the white strand. And the third all red strand forms a Hookstein pair. 4 DNA strands can also pair to form a tetraplex as shown here. But this occurs readily only for DNA sequences with a very high proportion of guanosine residues. As you can see here, guanosine can hydrogen bond among themselves, but they're all two hydrogen bonds and can result in the formation of a quadruple helix, or what is called as a tetraplex. This is quite stable over a broad range of conditions. Let us turn our attention to understanding RNA. An RNA is synthesized using a DNA template. A major difference between RNA and DNA is that RNA contains ribose instead of deoxyribose. Another difference is that RNA contains uracil instead of thymine. RNA is involved in translation. For example, messenger RNA, which contains the genetic information that is transcribed from DNA, is utilized by the ribosome to translate proteins. Transfer RNA is involved in carrying amino acids to ribosomes. Ribosomal RNA or RRNA is used by ribosome to detect mRNA. RNA is also involved in regulation. There are microRNAs, that are very small RNAs, in our cell. Short interfering RNA is used to knock down certain genes. There are other non-coding RNAs that are present in our body, and so much research is happening as we speak in understanding the function of such non-coding RNAs. mRNA or messenger RNA is a copy of DNA that is trafficked to the ribosome for protein production or translation. These mRNAs typically contain the information for only one gene in eukaryotes, but will often contain the information for multiple genes in prokaryotes like bacteria and archaea. mRNA is always single-stranded. RNA has no regular secondary structure but will fold into complex structures depending on the sequence. Shown here are two different mRNA sequences from bacteria and archaea. A single mRNA molecule may code for one or several polypeptide chains. If it carries the code for only one polypeptide, the mRNA is monocistronic, as shown here. If it codes for two or more different polypeptides, the mRNA is polycistronic. In eukaryotes, most mRNAs are monocistronic. But in bacteria and archaea, there are mRNAs that are polycistronic. Single-stranded RNA molecules tend to assume a right-handed helical conformation dominated by base stacking interactions. Now, these base stacking interactions are stronger between two purines, such as adenine and guanine, than between a purine and a pyrimidine. For example, guanine and a cytosine, or adenine and a thymine, or adenine and uracil in case of RNA here. Now, the purine-purine interaction that happens between two purines is so strong that a pyrimidine separating two purines, so if there's a pyrimidine that separates two purines, it is often displaced from the stacking pattern so that the purines can interact. Any self-complementary sequences in the molecule, an RNA molecule like this, produce more complex structures. Now, the structure that is shown here, as you can see, the bases are shown in yellow and the phosphorus backbone is shown in green. The color scheme is similar in this structure as well. And oxygens are shown in red, and as you can see, this representation is usually what is used throughout. When two strands of RNA with perfectly complementary sequences are paired, the predominant double-stranded structure, as shown here, is an A-form right-handed double helix. And this structure, this A-form structure, actually forms a right-handed double helix. However, strands of RNA that are perfectly paired over a long region of sequence are pretty uncommon, which means that you don't see extended double helical conformation of RNA molecules. The three-dimensional structures of many RNAs, like those of proteins, are complex and unique. Weak interactions, especially between bases, as shown here, base stacking interactions, are the ones that help stabilize RNA structures, as they do in DNA. Z-form helices have been made in the laboratory but not been observed. Now, the B form of RNA has not been observed as well. Only B form of DNA is available. If there is a double strand kind of conformation as shown here in this A form helix, this usually is caused by mismatch or unmatched bases such as this in one or both strands, and this is pretty common to see, like a bulge as shown here, or an internal loop, or a hairpin as shown here. Now, the paired regions as shown here in this single-stranded double helical conformation of an RNA, this generally have an A form right-handed helix, and this is a hairpin double helix as shown here. That this is the most common structure that you can see in an RNA. RNA molecules can have complex structures as shown here in this diagram. Now, this is a possible secondary structure of the M1 RNA component of the enzyme RNase P from E. coli. Now, as shown here, this RNA structure is very complex, and this is a single-stranded RNA. As you can see here, one strand goes throughout and pairs with the same strand that is coming up. Now, as you can see, there are brackets that are shown here, and these brackets, two square brackets, indicate additional complementary sequences that may be paired in the three-dimensional structure. The blue dots, as you can see, there are some blue dots, and this blue dots indicate non-Watson-Crick GU base pair. This is the GU base pair, which doesn't usually exist. Now, this GU base pair are only allowed when pre-synthesized strands of RNA fold up or anneal with each other. And there are no RNA polymerases that insert a uracil opposite to a template guanine. Please remember that. But these kind of unusual base pairing can be seen in complex RNA molecules. If you really want to understand the complexity of RNA molecules, and if you're really interested in understanding the structures, There is a game that you can play if you go to this website. And this was heavily advertised in NY Times. It says RNA game lets players find help a biological prize. And this game is very, very cool in the sense that it gives you an RNA structure and you can predict by base pairing various different bases as to what kind of secondary structures they can adopt. Please try it if you are really interested. Transfer RNA or tRNA brings amino acids to the ribosome during peptide synthesis, and tRNA carries their cognate amino acid that are complementary to the codons that are shown here. Now, how does tRNA structure look like? The three-dimensional structure of a tRNA is shown here. Now, this specific tRNA is a phenylalanine tRNA from yeast. Some unusual base pairing patterns can be found in this tRNA, as you can see here. One of the unusual ones is 7-methylguanine, which hydrogen bonds with guanine itself, which has a Watson-Crick hydrogen bonding with cytosine. Now, as you can see, this is a very unusual hydrogen bonding pattern, quite similar to the Hookstein hydrogen bonding, as we have seen before. There are other hydrogen bonding interactions, such as adenine pairing to N2-dimethylguanine. Again, this kind of bases are pretty unusual and can be found in RNA molecules. Adenine to uracil, which then the ribose of that uracil can hydrogen bond to an adenine. So these are some of the various different kinds of unusual hydrogen bonding patterns that can be seen among bases in RNA, specifically tRNA. Ribosomal RNA or RRNA are components of the ribosome involved in RNA recognition and protein synthesis. Ribosome has approximately 60% rRNA and 40% protein by weight, and it has multiple subunits as well. Large subunit acts as ribozyme, catalyzing peptide bond formation. So what are ribozymes? Ribozymes are RNA molecules that have catalytic activity. For example, a three-dimensional structure of a hammerhead ribozyme is shown here. The name hammerhead because the secondary structure at the active site looks like the head of a hammer. Now, this specific ribozyme is derived from certain plant viruses. Ribozymes or RNA enzymes catalyze a variety of reactions, primarily in RNA metabolism and protein synthesis. The complex three-dimensional structure of ribozymes reflect the complexity inherent in catalysis. And hence, the RNA world hypothesis is this: beginnings of life may have been self-copying RNAs. So it could have been RNAs that started the world or life.